Okay, sir. Good morning, students. Welcome to the last session of Unit 1, Diversity in the Living World, Chapter 4, Plant Kingdom. Today's topic is gymnosperms. In this plant kingdom, already algae, bryophytes, and pterophytes class has been taken, and I hope you understood very well. Now, gymnosperms. In this, you are going to learn about the pinus and cycas example of gymnosperms. Before that, I would like to appreciate the work of P. Prasuna, G. Nikita, G. Ankita, M. Varshita, S. Anoja, Sudipta, V. Srilata from Vangara TSRJC, B. Nirosha, Durga, Pooja from Hasanpati TSRJC, Asma, Grishma, Narmada from Vaira TSRJC. I also thank their respective JLs also for forwarding their names, and I hope all other children are also maintaining a separate notebook for the assignment work. When the colleges are going to open, you have to show it thoroughly to your teachers. So children, it's never too late. Begin if you haven't started it. And these children, they are doing their work exactly immediately after the classes are over. Sorry, in the afternoon session, immediately they are writing the work and forwarding and making me very happier. I hope the same thing will be followed by all other children in the different colleges. And complete your work. May not be your name is not seen here, but it will be seen on the next slide. The other teachers are going to keep your names. Forward the work to your respective JLs. Let us begin with the topic today. Gymnosperms. Gymnosperms are plants in which the ovules are not enclosed. Ovules are not enclosed. As you know, ovules are nothing but they are going to become the seed. So the ovules are not enclosed by ovary wall. And you know that ovary is going to become the fruit. That means there is no fruit formation taking place because of the absence of ovary and remain exposed. So the ovules are exposed. That means after fertilization also, the ovules are going to be exposed. That means both before and after fertilization, the ovules remain exposed. They are not enclosed in any fruit like structure. So that's why these are called naked seeds. So gymnosperms, they'll be having the naked seeds. The seeds that develop post fertilization are not covered, that is, they are naked. Gymnosperms are embryophytic. As you know that when seed formation is taking place, it is nothing but the seed is going to have an embryo inside it. The pollen grain and the egg cell both will fuse. Fertilization will take place. Zygote formation will take place. Zygote will be developing into an embryo. So this embryophytic stage can be seen in gymnosperms, and also these gymnosperms are also having tracheophytic. Tracheophytic means the vascular bundles are well developed. Xylem and phloem tissues. Archegoniate. Archegoniate means archegonia. The female reproductive structure is present. And phanerogams. Phanerogams means flowering plants. So in some of the books, sometimes it will be given as non-flowering, but don't get confused. Phanerogams means they are flowering plants. Here, the male and female gametes are formed by special structures called strobili or cones. The strobilus, it may be a male strobilus or female strobilus. This strobilus is going to represent the flower. So here we can consider these gymnosperms as phanerogams. They include medium-sized trees or tall trees and shrubs. So you can find medium-sized trees, tall trees, and shrubs. The giant redwood tree, sequoia, is one of the tallest tree. And ginkgo. Ginkgo is considered as one of the fossil. Why fossil? Because it is going to show some of the ancestral features. That's why it is considered as a fossil. Though it is living, it is considered as a fossil. And classification of gymnosperms by K are spawned in 1965. You can see here the gymnosperms are classified, and here you can see three classes. The first class is Cycadopsida, and most important in this cycadales, cycas plant you can learn in detail in this session. And the class Coniferopsida, it is having four orders here, and coniferales, cone-like appearance of the plant body, that is the pinus, you will be learning in this session. Class Nitopsida, and it will have an order Natales, where example is Nitum. Nitum is the example. So here in this session you are going to learn about cycas plant, pinus plant, general features, in order to get a clear understanding about these gymnosperms, what they are, how they are going to bear the strobili, and how the strobilus are going to give gametes. These gametes on fertilization are going to produce the next generation. So here a beautiful lovely picture of pinus tree is given, and you can see a strobili here. And this is a single strobili. That means it is a female strobili, because usually male strobili, they will be in cluster. They are born at the tip of the plant. So they are not at the tip of the plant. They are single. So it is a female strobili. Next, the plant body has a long shoot and a dwarf shoot. So the plant body usually it will have a long shoot, and this long shoot is having apical or terminal bud, and that is the reason it will be growing and growing. Okay, this is a long shoot, and it will be having dwarf shoot as well. Dwarf shoot will be given on the sidewards. Okay, dwarf shoots will be given like this. And the most important thing, the leaves, arrangement of leaves, it will be having scaly leaves, scaly leaves, scale like. So scaly leaves can be seen here on the complete plant body, long shoot. And these dwarf shoot, on this also you will be finding needle like leaves, foliage leaves. So scale leaves are there and foliage leaves. These are the scale leaves, and these green color are the foliage leaves. These foliage leaves are green in color because they undertake photosynthesis. And the most important thing about foliage leaf is that they will be having sunken stomata. Stomata are sunken because this pinus tree will be growing in a cool place where ice falling will be there, snow falling will be there, and water is not available in liquid state. That's why, to avoid the transpiration through the stomata, the foliage leaves, which are needle like, they'll be having the sunken stomata. So this is about the shoot as well as leaves. Long shoot, dwarf shoot, and scaly leaves and foliage leaves. So two types of leaves we have discussed here, scale leaves, foliage leaves. Scale leaves are brown in color, present over the body of the plant. Foliage leaves are given by these dwarf shoots which have sunken stomata. So long shoot has a terminal bud for unlimited growth, and dwarf shoot has no bud at the tip, so it is showing limited growth. The branches are in acropetal fashion, that is lower branches are long and upper branches are shorter, which gives a cone-like appearance. So in pine plant you will see the pine tree will be having the branches in acropetal fashion, the longer one at the base, older one, and the new ones will be at the tip region, and this is a cone like appearance. Okay, and they are in the coniferales, in the order coniferales, pinus. So this is a small introduction about this pinus tree, and we have to learn much more about this pinus tree. Root system we'll see in the gymnosperms. Most of the gymnosperms, they'll be having the tap root system, and you'll see adventitious roots also here. And you can see the most interesting feature, the corollide root. The most interesting feature, that is the corollide root. These corollide roots are nothing but a symbiotic association with cyanobacteria, that is the nostoc and anabena. These are the bacteria you know already, and these are photosynthetic bacteria, nostoc and anabena, and these are seen in the cycas for nitrogen fixation. So here you will be having the cycas plant. It is about cycas plant, corollide root. And this one is the pinus plant root system. Here another thing is there, that is mycorrhiza, the presence of mycorrhiza. As you can see, without mycorrhiza how it is looking, and with mycorrhiza. Mycorrhiza is nothing but fungal association with the root system. And in this fungal association with the root system, it is seen only in pinus. Corollide roots will be seen in cycas. These two are the important features about root system. You can see corollide roots for nitrogen fixation, seen in only in cycas. And association is between cycas root as well as cyanobacteria. And fungal association along with the root system of pinus is going to make the mycorrhizae, which is nitrogen fixing bacteria only, nitrogen fixing only fungus. Next, as you can see here, the beautiful plant body, it is showing the needle-like leaves. The leaves are needle-like. They are not flat. They are needle-like with as you can see, the brown part of the plant body having, and at the tip of the dwarf shoots, photosynthetic foliage leaves. These are the foliage leaves which you are seeing, green color. So two types of leaves, and these grow in cooler areas. Like in appearance, these have sunken stomata. It is monoecious. Reproductive structure will be. Okay, so this single plant will only give the male reproductive part also, female reproductive part also. So that's why it is mono, single, only one type of sporophylls. Male and female sporophylls means the reproductive organs are on the same plant. You can see the beautiful appearance of this pinus tree, and here sporophylls as third type of leaves. These are considered as third type of leaves. One thing I would like to discuss here about the common angiosperms which you see, which are having beautiful colorful flowers. The flowers are formed at the tip of the stem in bunch or single, whatever. And these flowers are nothing but they are the modified shoot. They are the modified shoot. Generally, the shoot will be having nodes at which the leaves will be given. Leaves will be given, and in angiospermic plants, common flowering plants, you can take an example. You'll see the flower formation will take place at the tip of the shoot. That means the shoot is modified. Flower is nothing but modified shoot. Why it is modified? If we take all the structures of a plant flower, the calyx, the corolla, the petals, the androecium part, and the gynoecium part. All these four, if we'll keep here on the tip of a plant or the tip of a shoot, at the nodes we'll find leaves. But here, for the flower, you'll see, if you just drag the flower like this, then you can notice at particular nodes only it is present. At the first node, you can see the green color calyx will be present, which is protective in nature, and beautiful petals will be present, and then androecium will be present, which is the male reproductive structure, and center gynoecium will be present. So just a beautiful flower appearance you can see in the angiosperms that you are not seeing in these gymnosperms. You can say that instead of the leaves on the shoot, flower parts are seen in angiosperms. Now, if we'll talk about these gymnosperms, where you are not seeing flowers, but the flower like arrangement is there. That is our sporophyll leaves. Okay, these are considered as a third type of leaves. Why they are considered as third type of leaves means they are also formed on the plant body, and they'll be having a specialized structure which is going to produce the spores. The spores are megaspore or microspore. Megaspore, it is in case of female reproductive organ. Microspore, it can be considered as a pollen grain, the male gamete. So here the sporophyll is present, and it is going to be in the form of a cone or strobili structure. And this cone or strobili structure are the sporophyll, which are considered to be the third leaves, or the strobili, which is going to represent a male flower or female flower. The sporophylls are two types, microsporophyle and megasporophyle. They form the strobilus or cone. And megasporophyll is the female strobilus. Megasporophyll, other name is macrosporophyll. Macro means large, the large one. And microsporophyll is the male strobilus. You can see the present at the apical region of the pinus. You can see for pinus, you can see so many cones are given. Okay, they are in cluster, they are in a bunch, and these are the male reproductive structure or male strobili or male flower. Now this is a female one, and you know the female one is not at the apex. It is on the sides of the plant body on the dwarf shoots, and it will be present only in single. And this is also going to get arise from the axil of the dwarf shoot. Axil of the brown scaly leaves will be present, and at the axil of this um, dwarf shoots only, you can be finding these female strobili. Female strobili, which is going to represent a female flower, is seen. So fixed the male flower, and the female strobili is going to represent the female flower. And this is a pinus tree. At the terminal, at the apical region, tip region, only you can see in groups. In groups, the male strobilus and the female strobilus will be on the dwarf shoots arising from the axil. Now here you can see the gymnosperm life cycle. You can see the sporophyte, which is the dominant stage having diploid condition. This is a plant body. This is dominant, that's why here, as in case of pteridophytes also and angiosperms also, you can see the sporophyte is the dominant stage in our gymnosperms. And these gymnosperms, you can see the dwarf branches, and you can see the long shoot. Okay, and these are branched or unbranched also in gymnosperms. Here you can see the branches are given here, and you can see the seed cone and the pollen cone. This pollen cone is nothing but the male gametophyte. The male gametophyte, the other name is microgametophyte. Where pollen mother cells are undertaking meiotic division, and that's why it is forming the pollen grains, which are the haploids. And the same thing is taking place in the female strobili. This is a female strobili. Okay, if you take one single strobilus, and if you are taking this mega gametophyte, this is also haploid, and this is going to give the egg cell, the female nuclei in the ovule. You can see the ovule structure here. This ovule structure, why it is written here seed cone means here only the seed formation will are female strobili. So here you can see both the male and female structures. This is a male structure and this is a female structure. This is the male gamete, pollen grain, and this is the ovule female gamete. Both of them, they are going to fertilize. When they fertilize, the zygote formation will take place. N plus N, it is 2N, the zygote. this zygote is formed here only in the seed cone. It is naked. As you can see, the undertaking mitotic divisions and becoming embryo. So this one is a sporophyte formation phase. Okay, and here the sporophyte is going to develop from this, and that is 2n condition, and it is the dominant phase. And this sporophyte again, as you know, it is going to be the male and female reproductive structure. This microsporangium, the male reproductive structure, megasporangium, the female reproductive structure, and they will be undertaking meiosis to produce the haploids. These haploids are the microspore, that is the pollen grain, and megaspore, that is a egg. This egg nuclei and the sperm or microspore, in simple language we can say the sperm and egg. Sperm is the male gamete, egg is the female gamete. This is a little bit smaller one, gametophytic stage, and this is a sporophytic stage. So here dominant one is sporophytic stage, so it is called diploid, diplontic life cycle, diplontic life cycle, or simply you can say diplohaplontic also you can say, diplohaplontic, because here many cell divisions are taking place and a gametophytic structure is formed here, and the zygote is going to form, so here haploid can be seen. So here diplohaplontic is the life cycle here for gymnosperms. Now coming to the next in detail about the strobilus look-alike. It is a male part. It is considered as the male flower also. So it will be in cone shape. You can see the cone shape. You can see the central axis. This is the central axis. This is the central axis. And here only you can see the microsporangia. This one microsporangia. This is another microsporangia. This is another microsporangia. So spirally it is arranged on the central axis. So you can see many microsporangia are arranged here on the strobilus, which is going to be the male flower and male strobilus. Here you can see one single number, that is the pollen sac. It is looking single from this view, lateral section, but from the top view, if you'll see, two pollen sacs will be there. Two pollen sacs will be there. For example, from the top view, it is going to look like this. And here two pollen sacs will be there. In this pollen sacs only, mother cells will be undertaking meiosis division, and they are going to form the haploids. Okay, by meiotic division, they will form the haploids. These haploids are only our the pollen grains, the gametes, which are going to form. I hope it is clear. This male strobilus is only the male flower, and this is a microsporangium, one single microsporangium. This is another microsporangium. So in this fashion, it is arranged in the form of a cone. Strobilus, that's why it is called as strobilus, male strobilus, rather than male flower. Male strobilus is referred here, and here you are having the pollen mother cell. So here from the top view you can see two pollen sacs in which meiotic division will take place, resulting in formation of the haploid gametes, the pollen grains. So this is very simple about pinus. Male strobilus produces spores. Male strobilus has a central axis over which microsporophylls are acropetally arranged. Each microsporophyll has a pair of microsporangia in which microspore mother cell undergoes meiotic division and forms haploid pollen grains. So the pollen grain, how it will be? The pollen grain is lightweight. It has a wing-like structure. If we see the pollen grain, this is the intine structure of the pollen grain. Exine will be somewhat wing-like. Okay, this wing-like stretched. It is for getting carried with the air. It will be helping for this pollination by wind. Anemophilous pollination means pollination by wind. This is a form which will be having a wing-like structure. That means this is a intine. Intine means inner part, inner cover, and this is a exine. Exine is somewhat stretched so that it can fly in air. Sorry. So here pollen grain germinates in the microsporangia. This point is very important. Pollen grain germinates in the microsporangia. Now is the ovary, ovule, stigma style ovary, you know that. And here they are going to germinate and form the pollen tube like structure from where the gamete is going to enter and fuse with the egg cell. But this is not happening. The germination of pollen grain is taking place in the microsporangia itself. Large number of pollen is released which has sulfur component, and that's why if you see a pine forest, there will be a yellow color cloud which is called sulfur cloud. It is called sulfur cloud. That is because of the pollen which are more in number. It is released at the terminal end only. It is present on the pine. The male pollen grains are released on the top layer of the plant body, and sulfur cloud can be seen. That sulfur cloud is nothing but the sulfur component is present in the pollen grain, giving it yellow color appearance. Coming to the female strobilus spine, and it is representing the female flower, and it is larger than the male cone. As you can, it is also having same appearance like that, only acropetal fashion only. You can see the ovule, but before that you can see the central axis here. This is the central axis, and you can see number of ovuliferous scales are there. These are ovuliferous scales. In this ovuliferous scale only, you'll be having the megasporangium. This one is a round one is a megasporangium. It is also two in number. From here, it is only one visible. But when you see from the top view, the ovuliferous scale will be having two megasporangiums. In these two megasporangium only, the ovule is going to form. Orthotropous ovule. Orthotropous ovule means you have learned in 10th class. Okay. This is, for example, an ovule. The egg cell will be present here and synergids will be present, antibodies like this. This is the chalaza region. This is a micropyle region, and the funicle will be down. So all these chalaza, micropyle, and funicle, these are in the same axis. Then it is considered as orthotropous ovule. And it is having single integument. Single integument means here two integuments you'll see for angiosperm, but it will be having only one integument. One integument will be seen. So that is a thing about the ovule which is going to form in the female strobilus. So female strobilus will be having central axis. On this you can see ovuliferous scales, and megasporangiums will be present. A pair of megasporangium. For each megasporangium you will be having one single ovule, not multiple pollen grains. It like pollen grain, it is having only a single ovule, and this ovule will be having the egg cell. And one more important fact about this one is carpellary scale, or you can also say the bract. Here bracts will be present. You can see the bract scale. These bract scale are very important. Why they are very important means they are going to hold the pollen grains for around 15 months. For around 15 months. As you have heard it clear, pollen grains will be coming and through the wind when they are carried on the Bragg scale, they are going to get deposited. They are going to get deposited in the structure. Around 15 months time it may be taking for the fertilization process to begin. And here when the fertilization once it has begun, after the pollination, fertilization, that is the fusion of male and female gamete will take place, then the seed formation is going to take place here only. It is also time consuming process. Seed formation as well as after pollination also, the pollen grain has to wait around 15 months time duration. And that's why the bract is present here, which is going to hold. In this, the lightweight pollen grains are going to get deposited, and the lucky pollen grain is going to fertilize this ovule, the egg nuclei, and the zygote is going to form here. And zygote is going to undertake mitotic division. It is diploid in condition. Because it is undertaking mitotic division, it is going to have the embryo, which is diploid only, and the embryo develops into a sporophyte. So this fertilization embryo formation is taking place here. That is nothing but the seed formation. These seeds are naked. There is no fruit here. They are open. They are visible. Naked seeds. And this is the thing about female strobilus. And next, central axis is surrounded by megasporophyll in spiral. There is an ovuliferous scale and a carpellary scale. In ovuliferous scale, a pair of ovules develop. The carpellary scale receives pollen grain, which remains for a period of about 15 months. Fertilization and seed formation is also time-consuming process. Seed in pinus has many cotyledons. One more important interesting feature is many cotyledons. The seed of pinus is having many cotyledons. And next, cycas. Another type of plant in gymnosperms we are going to learn is about cycas. This cycas plant, you can see, it will be growing in warmer areas, unlike the pinus, which will be growing in cooler places. This one will be growing in warmer areas. The foliage leaves are pinnately compound and photosynthetic. And you can see the pinnately compound leaves, and they are also photosynthetic. Scaly brown leaves cover the main plant body. Cycas is dioecious. Here, the most important thing is it is dioecious. Dioecious means male and female plants are separate. The male plant will be producing the male strobili, and female plant is going to produce the female strobili. And largest male and female gamete and largest ovule of plant kingdom can be seen in this gymnosperms. And in gymnosperms, the main highlighted part is they are going to show a largest male gamete and the largest female gamete, and the female gamete also, the ovule, is also the largest one. And wood of cycas is not durable. This cycas wood, as you can see, it is not durable because of storage of reserve food. That is the starch. Starch is stored in reserve in the parenchymatous cortex. The cortex is going to store the starch. That's why fungal infection, other infections may be taking place if starch is present, and so the wood is not durable. Endosperm is haploid. Endosperm, one more important interesting feature is generally endosperm will be triploid due to second fertilization. You can see in angiosperms, but in endosperm is haploid in gymnosperm and is formed before fertilization, and it is also formed before fertilization. Here a clear view of life cycle of this gymnosperm is also shown here. As you can see, as I said, the zygote is going to form, and this is the seed, and the seed is naked. You can see this is the embryo, and seed coat is one single seed coat. Only single integument will be there. That's why single integument, only single seed coat. And seed here, you can see it is having cotyledons, multiple cotyledons will be there. And finally, the sporophyte is going to form. The seed is going to germinate, and the sporophyte, the diploid sporophyte is going to form here. And as you know, the sporophyte will be producing the male and female cones. And these male and female cones, they will be giving the gametes by meiotic division. And these gametes, which are haploid, are going to undertake the fertilization, and after fertilization they are going to form the zygote. So you can see here the clear view of the microgametophyte. This microgametophyte, microspores, microgametophyte or the pollen grain. And here this is the ovule. This ovule is orthotropous ovule, as I said, and it is having only single integument. And this is the egg nuclei. And you can see the egg nuclei and the pollen tube sending the pollen nuclei to this one so that it can fuse with the female nucleus. And here, then what happens? The micro, then the fertilization is taking place and a zygote, diploid zygote is going to form. Next, haplontic and diplontic life cycle. Now, there is an interesting topic here, that is a discussion about the life cycle of these plant kingdom. If you see in the plant kingdom, you can also check the thallophyta, the algae also. The algae, they'll be having haplontic life cycle. Haplontic life cycle means the gametophyte is the haploid gametophyte, and it is the dominant stage. On this gametophyte only the male and female gametes are going to form, and then afterwards zygote formation will take place, and the fusion of male and female gamete, the syngamy will take place, and the zygote formation, which is diploid in condition. This diploid phase is very short, and immediately this zygote, it will not multiply, it will not become embryo, it is going to give the gametes here. It is going to produce the spores. It is going to produce the spores, and here you can see the haploid spores are formed here. And in this manner, you will see only the haplontic life cycle that can be clearly seen in algae such as Volvox, Spirogyra, and Chlamydomonas. So this is the haplontic life cycle. Now, if we further move and examine all others, just now we have learnt about gymnosperms, yesterday pteridophytes, and angiosperms. They will be showing this type of life cycle, diplontic life cycle. Diplontic life cycle means they will be having the sporophyte. Diploid sporophyte is the dominant phase, and this dominant phase is going to produce the gametes. Gametogenesis will take place. The male gamete, female gamete will form by meiosis, and because meiosis, it is forming the gametes. These gametes are going to fertilize, fuse, and the syngamy will lead to the zygote formation, which is diploid again. So from the zygote only the sporophyte will form. So here the dominant phase is sporophyte, and it is diploid in condition. That's why it is called diploid life cycle, diplontic life cycle. Instead of diplontic life cycle also, we can mention it as diplohaplontic life cycle also, diplohaplontic. Okay, or else simply we can say diplontic. Don't get confused with this important thing here. For algae, we are saying haplontic life cycle because the gametophyte is haploid, and this is the dominant phase. Okay, and here for pteridophytes, gymnosperms, angiosperms, will be having the diplontic life cycle because the diploid sporophyte, it is the dominant phase. And also you can see gametogenesis, which is having the sporophyte, which is having the gamete formation, which is haploid. So we can consider it as diplohaplontic also. But here we are mentioning diplontic life cycle only. That you have to remember, pteridophytes and all seeds bearing plants come under this diplohaplontic or diplontic life cycle, whatever. Now, the next important thing, that is haplodiplontic life cycle. Haplodiplontic life cycle, this can be seen only in the bryophytes. Yes, the bryophytes, where you can see the gametophyte. The gametophyte is dominant. The gametophyte is dominant and producing the male gamete, female gamete, and fertilization is taking place. Zygote is forming. This zygote is developing into a sporophyte, and that too, it will be present on the gametophyte only. It will be dependent on the gametophyte. Remember the food, seta, and capsule, the sporophyte. Then afterwards, the capsule is going to undertake meiotic division where the spores are formed. So these spores are diploid spores. Remember, these spores are diploid spores, and the diploid spores are going to give rise to a haploid gametophyte. So this is haplodiplontic life cycle. Why haplodiplontic means gametophyte is the dominant stage, which is haploid in condition. So haplo and diplontic stage can also be seen. So haplodiplontic stage, that can be seen in only and only bryophytes. And today's assignment is mention the fun characters found in cycas. What are the fun characters present in Saikas? Saikas also, the leaves will be curly in the beginning, then afterwards they are going to uncurl themselves, and archegoniate. And describe the important characteristics of gymnosperms. Gymnosperms, you have learned about the important characteristics, you have to write about them. This is your assignment. But before this, I would like to uh, thank here. I would like to thank our secretary, sir, for giving this golden opportunity for me. I wholeheartedly thank our secretary, sir, Sri Ramana Kumar Garu, for providing this platform where I connected with students of other colleges of this society. And it was an amazing experience for me. And I also thank AGO, sir, AGO and Deputy Secretary, sir, CS Prasad, sir, and coordinator of Botany, D. Sarala, ma'am. Sarla Reddy ma'am, she's working in Hassanpati, and she was always motivating me before the class and after the class also. Her feedback uh, made me do this work easier, in a easier way. And I thank all the children who forwarded their work to me, and all the best for your IP. Uh, Wish you all the best come to you. May you remain the same, enthusiastic in learning. Thanks one and all. And also, last but not the least, I also thank our technical team also. Thank you, sirs. End of the session. Sir, class is over. Sir, sir, class is over.
Completed, madam, class? Madam?